Hi everyone, we are very happy to present you our new work about the role of gut microbiome, biotin, metabolism and host biotin status in severe obesity, and we show how biotin and prebiotic supplementation actually can improve metabolism. So my group is working for many years on obesity, which is a complex disease which involves the alteration of organ crosstalks. And basically, among very important organs is the intestine, and inside the intestine, the gut microbiota. And thanks to a large European project called MetaCardis, we were able to study the gut microbiome of subjects at different stages of obesity. And we realized that there is alteration of gut microbiota biotin metabolism. So now I pass the word to my colleagues, first Eugénie Belda, who did metagenomic analysis on the data, and Liz Voland, who did all the mouse studies.
Thank you, Karin. So we have studied a good microbiome of more than 1,500 individuals of the MetaCardis study, where we have identified 4 main types of microbiome composition, including a particular one enriched in bacteroides lineages that was particularly dysbiotic in terms of microbial gene richness and microbial cell density. And this composition was increased in prevalence with the severity of the obesity phenotype. Bacteroides and firmicutes, which are the 2 main bacterial groups in the human gut microbiome, differ in their metabolism of biotin, with bacteroides being predominantly producers of the vitamin and firmicutes being transporters of the vitamin, needs to take the vitamin from the environment. So thanks to the availability of cell count data in MetaCardis, we have been able to compute the absolute potential of biotin biosynthesis and transport by the microbiome, and we have observed that this is significantly depleted in severe obesity, especially in extreme cases of dysbiosis like those of type 2 diabetic individuals treated by metformin. In parallel, we have observed also a systemic deficiency in the serum levels of biotin in the host in the context of obesity that was not explained by difference in nutritional intake. So we have done a set of mechanistic studies in mouse that has been done by Liz to elucidate the role of the gut microbiome in this metabolism of biotin.
So yes, indeed, Eugénie, we used several animal models to investigate these interesting findings. And first, we wanted to confirm that the production of biotin by the gut microbiota contributed to host levels. And we used 2 models, so conventional animals treated with antibiotics, but also germ-free mice, and showed that in these 2 models, the level of circulating biotin was decreased as compared to animals with conventional gut microbiota. Then we also fed mice with high-fat diets to induce obesity and observed a perturbation of the biotin metabolism, as we could observe in humans in severe obesity. And then we wondered how we could restore this metabolism. So first we looked at bariatric surgery and noticed that after the intervention there was an improvement of biotin metabolism, both at the host and bacterial levels, in humans but also in mice. And finally, we investigated a supplementation of a prebiotic and biotin in mice fed a high-fat diet and noticed that this strategy was efficient to improve the biotin metabolism, but interestingly was also able to improve the host metabolism because the animals receiving the biotin and the prebiotic gained less weight and had lower glucose alterations as compared to conventional non-treated controlled animals.
So thanks, both of you, and we hope that we convince you regarding the importance of microbiome biotin metabolism, of course, both for the gut microbiota per se, but also for the host metabolism.